Okay, this last part of Unit 4 is going to be the cell cycle. Now, I'm teaching our local biology class here, and I know if you've gone to school here, you've seen this before. So I'm going to kind of go through this, and we'll kind of make this a little bit more brief because you should have a basic understanding of this, but maybe we'll increase your understanding of it today and get you in. We're going to take it to a little bit more depth in this class. Okay, so just a review, okay? The cell cycle is very important to all life, okay? What's the matter? I'm not okay, I'll present. All right, I got you. How's that? It should be coming. Thank you, people at home. I'm going to need help till I get this figured out. Can you see me at home now? Tell me, can you see me at home? Okay, sorry, I didn't hear you. Okay. Anyway, back to this. Okay, so the cell cycle basically allows for reproduction, growth, and tissue repair. Now, human reproduction doesn't happen through what we're going to talk about today, but there are some cells that reproduce asexually where we do see this. Can somebody shut the main door over there? Thanks. So the cell cycle, what we're going to talk about today, is sort of the life of a cell from its formation until it divides. So it's a cycle because it happens in a circle and it happens over and over again, okay? Now, not every single cell stays in a state of constant reproduction the entire span of the organism's life. Brain cells and nerve cell are an example of this. Once they sort of get set up during development, they don't divide anymore. That's why if you injure your brain or nerves, they don't really regrow. If you sever your spinal cord, nerve regeneration is not something that your body does very well. So not every cell does this, but a lot do. Okay, before we go any deeper into this, we got to look at how DNA is organized, okay? So most cells, all cells have to package and organize their DNA before division, okay? This is almost like if you were going to move out of your parents' house, you got to pack stuff up before you create two households out of one, okay? So, what's that mean? DNA wraps around a protein called histone. So some of your key words here I'll start underlining, okay? So these are sort of like little packing boxes, and the DNA will wrap around them and condense, okay? This is all about making something. It's like if you had to carry around a 200-foot rope, okay? Dragging it around all over the place isn't ideal. So you're going to have to coil it up. Maybe, have you ever seen those things like you wrap the garden hose around? Think about it like that. You're going to have to coil it up, and the little bundles around histones are called nucleosomes, okay? I will say this is a very vocabulary thick unit, okay? A string of nucleosomes bundles up into something called chromatin, okay? Chromatin. So we have multiple levels of packing here, okay? Now, I want to warn you that there are a lot of C words in the vocabulary. There's going to be chromatin, chromatid, centromere. There's lots of C words, so try to keep those chromosome. Try to keep those C words straight in your head, okay? So when the DNA is all packed up like this, it's not easy for ribosomes to get to it and copy proteins. So this is not a state that the DNA is in all the time. So look at this section here. when the cell's not dividing, it's not condensed. Just like if you're just going through everyday life, you don't have your clothes in a suitcase, okay? So this is only during certain periods in the cell cycle that we bundle all this DNA up, okay? So after DNA replicates, we get what are called chromosomes. And it's sort of like its final form, okay? It's all densely packed up, and we see basically these classic sort of like chromosomes kind of look like this right here. You've probably seen like X-shaped chromosomes. Okay, that's actually 2 of them, but just want to give you an idea of what they visually look like. Now we're going to show you lots of diagrams today, but all right. So once we are into this chromosome form, we make a copy of it, okay? And that copy lines up with its pair, okay? And where does it line up? It lines up in this place called the Centromeric Region. So chromatids will have this little sort of bundle in the middle called the centromere, okay? And that is where they're going to sort of connect together and hang out. So what are these 2 things called? These are called sister chromatids. It might be a better idea, now nobody calls them this, but in your head I want you to think of them as twin sister chromatids. So they're identical, okay? So if this is the gene for some particular protein right there, the exact same gene will be for that same protein over there, okay? So they are exact copies of each other. They code for particular proteins, and they are going to be held together in the middle by this little centromere. Okay, another C word. Okay, so centromere is the spot where they are closely attached. Okay, and I always think of them as being like a mirror image of each other. Centromere. It's the center of where the mirror image is. Okay. All right. Next thing I want you to understand is kinetochore. Kinetochore is the spot where they actually are going to connect to these little tow ropes called Meiotic Spindle microtubules. That's a lot to say, but this is where the spindles, okay, are going to hook onto them. So it's sort of like a spot. You can kind of see these little cat whiskers right here in this area right here. Those go way off the page, okay? Those are going to be attached to some areas that pull these apart, okay? So this is all getting ready to divide. So right now, we're really just looking at terminology, okay? Lots of terminology coming at you really fast. Might want to make some flashcards or review or notes or whatever with all these terms, okay? If we go back, look at all these new terms. I got 1, 2, 3, 4, 5, 6 new terms, and we're just getting started, okay? So this is one of those things you probably have to look at more than once, okay? That's why I record this stuff. All right, so here's sort of visual form. You're going to start with all the way down here, unwound DNA, okay? It's going to wrap up on these little things called histosomes, which turn into nucleosomes, which turn into chromatin, which becomes a chromosome, and then those become sister chromatids. Okay, so lots of terms here in order, and all those start appearing in the nucleus. Okay, so remember, this is a packaged form. DNA isn't really usable by ribosomes in this point, and we're getting packed up and copied so we can divide the cell. All right, how are we doing, everybody here and at home? Everybody staying with me? I got a thumbs up here. How about a chat at home? Everybody okay? Can you see now? Brian, thank you. All right. Lots of new terms, but this is stuff you've seen before. How many of you remember, like, yeah, I've seen a little bit of this? Okay, I'm getting lots of yeses. Okay, good. Now, just a little talk about genetics here. Just a genome is everything, all the information in all of an organism's DNA. Okay? That's going to be represented with multiple chromosomes. And then down in the chromosomes, we may have genes for certain proteins. So don't mix up gene and genome. Again, new terms. The genome is the whole thing. That's the whole library, okay? The gene is sort of like an individual chapter, okay? It tells you, here's the protein to make whatever, okay? So just to review that, a genome is everything in the entire cell's genetic makeup. Now, just to review, we talked about this already. In prokaryotes, that's going to be a single circle of DNA. In eukaryotes, it's going to be a linear chromosome. Okay, so usually we see those kind of like that. Now, every eukaryote has some number of chromosomes that's standard. In humans, it's 46 or 23 pairs, okay? In chimpanzees, it's 48. In elephants, it's 56. There is no correlation between the number of chromosomes and the complexity or the evolutionary state of the organism, okay? There are ferns that have over 1,000. Humans have 46, so I'd like to think that most of us are more complex than a fern, okay? So, I don't know, I've met some people with a personality of a fern, but we really don't want to get into like, well, it has more, so that means this. It's arbitrary, okay? And some of that has to do with how the reproduction of especially with the fern, how that happens. But there is no real connection between these, okay? All right, everybody clear on genome? Prokaryotes have a loop. Eukaryotes have linear, okay? So we'll go ahead and move on. All right, a little bit more about this. Homologous, okay? Homologous means the same, okay? So you have 23 pairs of chromosomes. This is like having 23 pairs of socks. Now, homologous means the same, but these are not exactly the same, okay? It's almost like having a pair of socks with print on them. They're usually not exactly the same, okay? What is the same on them is, let's say this is the first chromosome. You have one copy from mom, so I'll draw your little female symbol there, and one copy from dad. Okay? Usually dads are blue, but I went backwards this time. So, on that chromosome, dad may have given you a gene for, now I want to stress this, eye color is controlled by more than one gene, but let's just simplify it for a second. Dad may have given you a gene for blue eyes, and mom may have given you a gene for, I don't know, we'll go brown eyes. I know the colors aren't working out here. Those are the same gene, but they have a different genotype. Or it could be the same, okay? So maybe down here, you have the same. Now, if you're kind of like, yeah, I remember getting one allele from mom and one from dad. This is where like the big B, little b comes from, okay? Maybe this is a D and a D, okay? A little louder. It depends. Usually in high school biology, we study very simple types of dominance. But yeah, usually one is going to override the other. That doesn't always happen, okay? But usually it's one overrides the other. Sometimes it's a mix. All right, remember the flowers, red and white would give you pink? Yeah, okay. All right. So the big deal right now is each chromosome for this homologous is going to be carrying the same informational areas, but not necessarily the same information, okay? That's the big deal right now. You need to know homologous chromosomes, they carry the same information set, but they might have different information on it. A way to think about this is, in Ohio, everyone's driver's license has the same information in the same places. Okay? There's a place for your picture. There's a place for your name. There's a place for your weight, eye color, and all that stuff. And it's the same on every driver's license, what's on it. But there's different information. Okay? So think about it like that. Homologous means everything's in the same spot, not everything has the same info. All right. During replication, you get a copy of dad's and a copy of mom's. Okay, so we're going to split these up later, but we have to copy before we split. So that's what we've done with the sister. So the sister chromatids, they are identical twins, okay, they're identical twin sisters, but they're getting ready to copy homologous pairs. All right. So, there is a practice FRQ on Schoology that's this exact question. Just so I know you're out there, jump on. I'll give you some points for answering this in class, okay? It has to be done right now. I'll grade it right afterwards. Jump on to Schoology. We'll take a little pause so I can catch my breath. I'm going to pause the recording. You guys go ahead and answer this on Schoology. All right, we're picking up the second half of the recording, just talking about the difference between somatic cells, which are body cells, and gametes, which we'll talk about here in a second. So we were saying that these divide by mitosis, okay, which are the type of cells in your toes, if you're looking for a way to remember that. Now, in humans, that number, the diploid number, is 46. 23 pairs, half from mom, half from dad, okay? And that's why when you make a Punnett square, the letters come in pairs, okay? Big B, little b. Mom B, dad B, okay? So that's kind of how that's going to work out. Now, gametes are reproductive cells. So the only gamete cells in humans are sperm and egg, okay? So these are haploid. They have half the number of chromosomes. They're only going to have 23, okay? You cut them in half, you put them back together. That's the birds and the bees right there, okay? These divide by meiosis instead of mitosis. A little overpronouncing there so you can tell the difference. Okay, so the only place these cells are dividing are in your reproductive organs. So the chromosome number in there is 23. It's half of what it is the body cells. That's why they're called haploid, okay? So this unit right here that we're in right now is going to focus on this side over here, okay? We'll do this a little bit later, okay? So don't, for now, it's not like I want you to forget about this, but this is what we're going to focus on over here. All right. So hopefully you've seen something like this before, okay? This is the cell cycle. It's a circle for cycle. And we basically have a very little section of this that we're focusing on right now. Most of what we're focusing on right now is the interphase, which is the longest part. Mitosis is actually a really, really short part. Now, cells can go through mitosis very, very quickly. Like if a plant is growing very fast, they can stay in mitosis, but most cells, once the growth stops, they're going to stay in this big orange part here we're going to call interphase. So we will break all this down. Okay, so cell cycle basically switches back and forth between interphase and mitosis when the cell is growing. So here's the general layout. You have G1, S, G2, mitosis, and then cytokinesis, which we're going to break all these down, but these should be vaguely familiar to you if you've taken a biology class in your earlier years of high school. Okay? So interphase is the biggest, longest phase. It's broken down into these other phases that we're going to talk about. Okay, so let's break down interphase. Interphase is the longest portion of the cell cycle. Okay, most of the time the cell is going to be parked in interphase. So we're going to divide interphase down into a couple different sub-parts. Okay, G1, this is where, think of the G as growing. Okay, so this is where the cell's just growing, doing its normal thing. Okay, maybe this is a skin cell. It's just sitting there protecting you from the environment. Okay, maybe this is a glandular tissue that makes something for your body, like your saliva gland or something like that. Just going to sit there and pump out saliva. Okay? S would be the synthesis phase. Now, what is being synthesized in the synthesis phase? Okay, DNA. Synthesis means to make, so we're making DNA in the S phase. Okay, DNA synthesis is happening in the S phase. Okay, so this is where DNA coils up into chromosomes and you make a copy. So the cell in S phase, the cell is very busy getting ready for something to happen. All right. After synthesis, we add a second gap. Okay? This is the final stage before mitosis. Okay? Final growth. It's getting ready for mitosis. All right. The M phase, this is mitosis. We have the nucleus dividing. Then we have cytokinesis. You guys see cytokinesis at home? I think I might be a step behind. There it goes. And cytokinesis is the rest of the cell dividing. Okay, so think of mitosis as a special nuclear event and cytokinesis as the everything else. Okay, so this is the all right. Pacing okay, everybody? All right, good. Okay. So what's the end result? Okay, we have two identical daughter cells, okay, and those cells are diploid, which means they have a full set of DNA. Okay, they've got a mom gene and a dad gene. So these are diploid. You want to maintain the same amount of DNA in all your body cells. We don't want to cut it in half every time. That would be bad. All right. So let's focus only on mitosis. The last slide was interphase. Now we're going to zoom into mitosis. This is what we're mainly concerned about. Okay, I know these pictures might be a little hard to read at home. Okay, but we're going to go through them a little bit closer, step by step. Okay, mitosis is divided into 4 main phases, but there's sort of like a subphase or like a minor phase we're going to throw up here today. Okay. So we're going to have prophase. You've probably heard this abbreviated PMAT. Now think of it as PPMAT. All right, so prophase is sort of that getting ready phase. We're going to break all these down individually. So here they are, okay? You have prophase, pro-metaphase, which is kind of like a getting ready for metaphase. Then you've got anaphase, telophase or telophase. I've heard it pronounced both ways. And then you've got cytokinesis kind of happens simultaneously, okay? So let's go through each one of these individually. I'm going to try to get through this today. I don't want to push every day like this, but I'm going to go a little bit hard today. Okay. So, first of these phases is prophase. Let's look at what you see. Okay. What are the big key events of prophase? Okay. This is a get ready phase. Okay. The chromatin condenses. Okay. This is basically, let's pack for the trip. This is the DNA coiling up into chromosomes, and you see the nuclear membrane start to break down. So we're going to see these sister chromatids start to appear. So normally we would see them like this. Okay, but now we're going to see them like this. All right, so the other thing we see are meiotic spindles. Okay, so meiotic spindles are these things right here. Let me see if I can. These sort of rope structures forming. They're getting ready to think. I always think of them like tow ropes, okay? I'll give you a big break at the end. I know the period just, the bell rang. We'll keep going. I'll give you a big break at the end. You also see another C word, okay? And that's centrosomes. They kind of look like little pop cans, okay? Alrighty. So, moving into what's called pro-metaphase. Okay, kind of like we're almost to metaphase. The nuclear envelope completely breaks down into fragments, okay, and that will reform later. So we don't really need a nucleus anymore. The membrane we need to get rid of it. Okay, so microtubules shoot into the nuclear area. So what does that mean? That means that these guys start finding attachments on the centromeres of their sister chromatids. And they call that area the kinetochore. There's so much new terminology. You just kind of have to take some time to study it. Now, they call these astral microtubules. What's that thing on the keyboard when you hit Shift 8? An asterisk, okay? So astral means star-like, okay? So astronauts, astronomy, okay? These things look like stars because they have all these lines coming out of them. All right, metaphase. In metaphase, here's how I remember metaphase. Everything lines up along the middle. Everything lines up along the middle. Well, what are the everythings? Those are the chromosomes, sister chromosomes. Okay, so the centrosomes, which are the little pop cans, they are at the north and south pole. Now, it doesn't really have a north and south pole. That's just me drawing a picture. And all the chromosomes line up on the equator. Okay, so middle, meta, okay? And what we see here, we call this place they line up, we call this equator the Metaphase Plate. Okay, it has a name. But I like to think of metaphase as middle. That's how I remember it. Now, at this point, we have microtubules extending out from the centrosomes to the kinetochores. Okay, that's the spot right here. These are the kinetochores here. I drew a little K on it. Everybody okay? I know this is a lot to take in one day, but I kind of really want to get rolling on this. I think we can actually finish the year on time if we push a little hard. Okay, anaphase. My key word for anaphase is chromosomes are moving away from each other. The other thing that I think they kind of start bending like this, and they look like letter A's without that bar there. Okay, so they're missing this bar right here, but they basically look like halfway drawn letter A's. Okay, let me back that up a little bit. Okay, so A for away, but they kind of look like letter A's. They're just missing their crossbars. Okay, so they start pulling away from each other. So that's how I remember anaphase. Okay, hopefully you'll start picking up on some of these little memory tools. Sister chromatids separate. Well, separate, another word for that is move away. And they move to opposite ends of the cell. They migrate north and south. Okay? And what's happening is these microtubules are shortening. Okay? They kind of are like little tow ropes that pull them apart. So you get this tug of war where they snap apart. Now, I want to ask you some concept questions. Let's say these are the two that snapped apart. Is the DNA in them similar or different? Somebody answer that. Similar. They're exact copies. Okay? Very good. Very good. All right. The cell elongates, okay? Instead of being circular, it's going to get more egg-shaped. It's going to stretch out. Okay, we're going to start seeing it get ready to turn into two cells. All right. Telophase, okay? The big thing I like to go with the T here is there are now two cells, okay? And they are starting to cut in half, okay? You get this thing called a cleavage furrow right here. Okay, that should be easy to remember. Now, cleavage just means cut. That big square knife a butcher has is called a cleaver. Okay, so cleavage just means cut if you're wondering what that means. All right, so what's going to happen? Two. Telophase is two. Telophase is two. Telophase is two. Okay, so you get two daughter nuclei. Okay? And a nucleus starts forming again. Okay, you get your own nucleus now. Chromosomes are going to unpack a little bit. Okay, they're starting to arrive. They're kind of like, I've reached my destination. We can unpack. That was probably too fast. Okay. So a couple things. This is where we have a little bit of a difference between plants and animals. Okay. In animals, we get this thing called a contractile ring, which is just a ring that contracts and closes down. Okay. All right. And that's going to be driven by something called actin filaments. Those are found in muscles. They kind of move things around in cells as well. In plants, you get what's called a cell plate, which reforms the cell wall. All right. Also happening right now is you're copying all the other organelles. We don't really talk about those right now, but you're going to get another mitochondria. You're going to get another, Golgi body, another, lysis. All that stuff's going to also start copying. Okay. They are. Yes. I'll just tell you this. They both take ATP to create motion, but this is basically a ring that's sort of cutting it in half. It's almost like if I put a string around some jello and closed it up, it would cut it in half. That's kind of what's happening here. Yes, okay. They're done, and you're going to see them fade away. Okay. So one of the things we want you to be able to do, and I kind of asked you the question there, okay? Remember, you have a copy of every gene from your mom and dad. So you have 2 copies of every gene. Somebody brought up, aren't sometimes one of them dominant? Yes, sometimes one of them is going to be expressed over the other one. Let's not worry about that too much right now. But in the future, when we say there's like a big B, little b, this is where they come from, okay? So I want you to draw that connection now, but let's kind of put that on the shelf for the moment. So we got a red chromosome from your mom. We copy it, and then we take 1 to each cell, and then the daughter cells each have their own copy. And if you look at the end results, these are going to be equal, okay? So when we go this way, okay, we're trying to get back to where we started. That's why we call it a cycle. And so the idea is to get to the same place. On test questions, if it's mitosis, these should match, okay? If they don't, something went wrong, or they're trying to give you an answer that's not right, okay? So to get 2 copies, you have to make a copy. So right here, we got our copy. Then we're going to just put things in the right spot right here. What phase does this happen in right here? That's S phase, DNA synthesis, okay? All right. So the parents, you always have double the number. You have 2 copies of every gene. That's what that 2N means, okay? After S phase, which I just told you about, you have double the amount because you're getting ready to make a copy. 2 chromosomes, 4 chromatids. Okay, it's kind of lots of terminology to keep straight here. Okay, and a lot of it sounds the same. Okay, and anaphase, now we have 4 chromosomes. Sorry. I would say this is one you should be able to pull apart pretty readily. This is something like every student in the class needs to have a good handle on. Okay, so this is just like how many are there in each phase, okay? You should know how many chromosomes there are based on what's going on. So if you can visualize this, you should know how many are there in each phase, okay? How many copies are there in each phase? So in mitosis, there's a lot of times where you have 4 copies of each gene, okay? But they're going to get segregated out, and you're going to have your mom copy and your dad copy end up in each daughter cell. All right. So we're going to do a little review right here, okay? What phase is this right here? Metaphase. How do you know? These guys are all lining up on the middle. Okay, so that's your metaphase. What's this right here? Anaphase. They look like little A's and they are pulling away. Okay? What is this one right here? Looks like it's in the middle of well, cytokinesis slash telophase, okay? They're going into the, but both happen simultaneously, okay? What would you say this one right here is? I thought I heard a prophase out there, and that would be right. Now, one thing that's important for you to understand as biologists is these are not like clunk, clunk, it goes to the next stage. It's not like clunk, it happens gradually over time. So you may hear people say like, well, that's early metaphase because they're not quite perfectly lined up. Or maybe they're just starting to pull apart and some of them are still metaphase and some of them are just starting to pull away from each other. So maybe it's like late metaphase, early anaphase. So just be careful. Like it looks like this one is late metaphase heading into anaphase, okay, compared to this one, which is like very clearly in metaphase. So this is M-A-T. I'm probably missing some here. This one's in telophase right here. Look at this one right here. Okay, 2 nuclei. That's probably an early telophase. I'm probably missing some, but those are the ones that are easy to see. Okay, those are the ones that are easy to see. All right, let's jump. Let's try these questions right here. Wait. Let's not try these questions. I have these questions on Schoology. There are only 3 of them. They're in the packet. Let's go ahead and call it a day, but answer those 3 questions on Schoology. I'll give you a 15-point quiz grade for those, so they're obviously open book. So go get those done for me, and we're going to go ahead and call it right here, okay? So get those done, and you have about you've got about 20 minutes to get those done, then take a little bit of a break before your lunch. I'm really trying to watch screen time and build in breaks, okay? I just feel like, especially people at home, there's only so much screen you can set and watch before this goes. like ZP over there said, basically it's tough to sit there and absorb it at home. He was saying that he didn't get much of what was going on at home. So I'm going to go ahead and stop. I'll get this posted, get your 3 little review questions done. I'll keep picking away at grading your projects. Let me go ahead and stop the recording.